Okay, let's start. Good morning, everyone. In the last class, we have discussed about. Okay, you cannot see the screen yet, right? Okay.
Yes, sir.
Yes, I'm sharing now. Okay. In the last class, we were discussing about melanoma stem cells. We learned about melanocytic stem cell that is responsible for producing melanocytes. Melanocytes are important, and we'll know about the several layers. Fat layer, there is dermis, there is epidermis. Somewhere around here you will find these melanocyte stem cells, melanocytic stem cells, which will give rise to melanocytes, these, and they eventually will form the keratinized layer, and then they'll be worn out or they'll be eroded. So somewhere there could be a possibility that a mutation may occur, because of which melanoma may arise. And before going there, we have to also discuss about what is melanin. Melanin is a UV protectant. The more melanin we have, there are different two other pigments also. The more melanin we have, the darker our skin pigmentation is. They have several functions in immune, metal chelation, endocrine functions, and so on. But they also mutation might result, in because of which neoplasm may happen, that is uncontrolled cell growth. And then we will see, because of once a mutation happens and cell cycle is going berserk, that means the rate of replications or cell divisions is increasing, which means there is more probability of mutations, which means there is more probability of getting more traits or phenotypes, which will make it more aggressive, right? So in a state, once a mutation has occurred that caused cell cycle dysregulation, then everything else goes, and then neoplasm, and eventually some of the cells might gain mutations that would have allowed metastasis. Metastasis is the process where it will go into the bloodstream and from there move to many other places. Melanoma is also one of the problems, aggressive in some sense, because if you notice that the skin has a lot of mobility, the skin cells have a lot of mobility. As I said, when they cell when the skin or a person becomes fat, they might become the stretch marks. There is a lot of stretching, and if there is injury and this part of it is gone, then skin cells will keep growing into. That means inherently there is some migratory potential within the skin, and the skin is covering the whole of the body, right? So there are inherently motile in a way, and then if mutation occurs, they can become really aggressive and move faster to many other or metastatize. We also discussed about some of the cell surface markers and other functions and what may happen. And what we are going to discuss some of the regulation today. Before going to the regulation though, we are going to talk about what is it to be a normal melanocyte stem cell and what is it to be a malignant melanoma stem cells. Melanocyte stem cell is the normal one, okay? That is, you can consider until here, this is all normal. And in the bottom part, we are discussing about the malignant or cancerous, malignant things, right? So melanocyte, it has several signaling pathways like north signaling, wind signaling. That is what we are going to discuss today. And they produce E-cadherin. If you remember in cell biology that you may have learned, E-cadherins will allow it's like a zipper thing where two cells producing E cadherins will bind together and will remain in contact like that. So that is um, what the melanocyte stem cells will give rise to. Melanocyte stem cells will give rise to melanocytes and some other cells which may give rise, which may form, which may express E cadherin and will remain, or will try to make connections with other cells producing E cadherin. When's a neoplastic transformation? Here, transformation is always, when you talk about eukaryotic cells, transformation means becoming cancerous. That means because of some mutations, the melanocyte stem cell, or it could be intermediate cells or transit amplifying cells, may become malignant melanoma stem cells. And then there are so many things that it's unclear, but what we know is they produce, there are a lot of signaling pathways, including VEGF, which is important for angiogenesis, and they start producing N-cadherin, that is neural type, and then they become malignant. They create malignant microenvironment, and then they keep spreading around. So how do we identify malignant melanoma stem cells? It is the same redundant thing that we have already been discussing. One of the things to characterize whether they are melanoma stem cells or not is that you can use malignant embryonic stem cell medium and put the cells in. Say, how do we select them? You take a culture medium, and the medium is human embryonic stem cell medium, HES. And then you put, say, if you had stem cells and normal cells, the normal cells usually die when you put in HES, human embryonic stem cell medium, because of abundance of mitogens. Mitogens are those factors that will influence the cell or coerce the cell to divide. Differentiated cells, okay? These are, when I said normal, I meant differentiated cells. They usually die when put in high concentration of mitogens. But what we are remaining are the stem cells. Say if you are putting a sample that is of a tumor biopsy, which will consist of cancer stem cells and some of the differentiated cells, and if you put like this, the cancer stem cells will survive and the rest will die. And that is another way of how you can identify or select the cancer stem cells. The other one is forming spears, and that is one we have already discussed in the previous class. One of the ways is to allow only cancer stem cells can form spears, which are something similar to that of embryoid bodies. And then there is one more, is using FACS, or based on the cluster designation CD20 plus and CD133 plus. Okay. So what are the properties? Again, this is redundant. To start with, there are two properties that are responsible for cancer stem cells. Cancer stem cell, right? There are two properties to be for stem cell. One of it is self-renewal, and that is here. Self-renewal is a stem cell-like property, and there is one more, that is differentiation, right? To be qualified as a cancer, we also discuss as tumorigenicity. This is for tumorigenicity, right? Those are the three properties for every cancer stem cell. There are also some other things that we need to discuss is they are usually resistant to chemotherapy or radiotherapy, mostly because they are quiescent. That is one of the people think that it is a quiescent. That is why it is the They are resistant to chemotherapy or radiotherapy. And this is an efflux pump. Say if we put in some chemotherapy, it has a ability to efflux it. That is one of the ideas people think of it. So, and immunomodulation, somehow these cells can coerce the immune system that it is normal kind of thing, and because of which immune system cannot kill this cell. So these are some of the mechanisms of resistance. And as we discussed before, if there is a tumor or cancer, we treat with radiotherapy, chemotherapy, and other things. Majority of the cells die, and after 1 or 2 years, again there is relapse. That is reformation of the tumor. And that is because of these resistances. And resistance could be several things. Somehow, even though we make several damages, the cell has impaired apoptosis, so it does not die out. Okay. Yes. And that is what will allow the tumor progression and many other things. So the central properties you should remember for every cancer stem cell is, first thing is tumorigenicity or proliferation. That is the cancer causing property. And the second and third are for stemness, that is self-renewal and differentiation. So while we are discussing some of these, we are supposed to also discuss 3 signaling pathways. So this you can consider it as a part of, if you are reading, it's a revival or revision of signaling pathways that you might have learned in other courses. Notch signaling, hedgehog, and Wnt signaling. I think we will discuss about notch and Wnt signaling today. This is important in 2 ways. These 3 signaling pathways, Wnt, sonic hedgehog, and notch, they are implicated in, or they are involved in normal stem cell behavior. There are also other ways where other cell types where these signaling pathways are a function. And at the same time, you can also find these signaling pathways that are also involved, or because of dysregulation or overexpression, some form of they are involved in tumors as well. So if you look at the hematopoietic, epidermal, and gut, the Wnt signaling is involved, and it is also involved in colon carcinoma, epidermal tumors, and so on. So this is normal, and here we are talking about cancerous ones. Similarly, sonic hedgehog signaling, hematopoietic stem cells, neural stem cells, or progenitor cells, and germline. They are involved in the self-renewal of these cells. And the same is the case also with cancers of the medulloblastoma and so on, right? So what I want you to draw is learning about signaling pathways is important in twofold. One is to understand the normal stem cell, how these signaling pathways are involved in it, and at the same time, how, if dysregulated, how can they be, or they are involved in, or in cancerous phenotypes. So I will send these links. There are some of the links. You can also find many of them on YouTube. You can go ahead and check it out. And some of them are very good explanations. We will not go into all the depth of it, but we will go into as much as we require to understand how the connection between what is the signal and what is the eventual consequence of that signaling pathway. Before going, I would want to have a general discussion, as I was saying. And prior to that also, I want you to realize what makes a cell, its form and function. The cell is constantly receiving information from its other cells. signals. Some signals will bind here to cell surface receptor, region receptor, for example. Some is by physical contacts with surrounding cells or extracellular matrix. The way a cell is surrounded, the whole three-dimensionally, okay? In three-dimensionally, how the cell is surrounded and the interactions, the chemicals it is receiving, and there it can be also mechanical forces, right? Everything is, the cell is able to, is getting signals from all these sources, and that is eventually going to influence the nucleus gene expression, and that is how the response will come, and eventually the cell may change its shape, morphology, depending on the need. So when I said there are several signaling pathways, each of them is giving a signal. There will be several intermediates, and there will be, in each of these you are getting, and there is usually crosstalk between signaling pathways as well. So if you want to put into perspective the function and form and function of a cell are cumulative outcomes of all signals, including physical. From distant, you can have a hormone coming in from a very distant place, or physical contacts from the nearby cells, or mechanical forces, many things. All signals received. And there is one more thing, and inherent genes expressed, right? That is what you can say. The form and function of a cell are cumulative outcome of all the signals received and inherent genes expressed. Say, for example, this is a signal molecule, and the receptor for it is not expressed, then the cell cannot respond to that signal, isn't it? So the cell, you call it as competence also, in a way, that the cell should be able to receive the signal and then respond to the signal. So there are inherent capacity, inherent properties, and the external ones, signals coming from outside, and what inherent properties it has. Can it respond to the signal or not? Okay, keep those things in mind as a system. Cell is a system, it's not an isolated part. In this lecture, we will learn a few things as if these not signaling, hedgehog, and so on, we will learn them discretely as separate entities. But I think at one point, we will have, directly or indirectly, there will be some influence. And at the level of cell as a system, it's all integrated things. Okay. So first thing is, a cell would receive a signal. Signal can be in the form of, it can be in the form of a chemical, a mechanical force, or a physical contact. And eventually it is going to influence the gene expression, right? Not everything should result in gene expression, but majority of them do. So for the signal to be detected and any consequence that should happen within the cell, we will need a receptor, right? Only then the cell can receive the signal. Receptors can be on the cell surface, or they can be in the cytoplasm, or probably also in the nucleoplasm. For those peptides or other compounds that cannot cross through the cell membrane, we usually have cell surface receptors. And for those that can cross the cell membrane, you can have it in the cytoplasm. And this is the signal. We have reception, and there should be some compound, some molecule that actually does the work of receiving the signal. I mean, signal is received that should influence the gene expression. And that component, I call it as the doer, the one that performs the action on the genes so that gene expression can be altered. So eventually, also the doer, the molecule that has to be that performs the action or influences the gene expression should be regulated. Primarily, there are two ways of how regulation is maintained. One of it is by stability. That means usually this is the door is not stable, but if it becomes stable or abundant in the cell, and then it will go into the nucleus and perform an action, that is influence the gene expression. There is another form of regulation is the doer is produced but it remains inactive because of a post-translational modification. The doer then becomes active and then allows the gene expression or modulates the gene expression. That is how this cycle is. So we need a lot of regulatory things, right? We will see. Say a signal is produced, and if it is constantly produced, then you will have constantly upregulation of these genes, and that is not good for the cell. So we should have a way of deleting the signal too, right? Once a signal has come, the production of signal, the degradation of signal, both are required. There will be. Otherwise, if once a signal is produced and it never goes off, then it is harmful. These genes, some of which will be kept turned on all the time, and it's not good for the cell. So there will be mechanism. There is a half-life for every signal in a way, and that is the degradation represented here. Similarly, for receptor, we should have had the expression of the receptor, that is production of receptor. At the same time, we should also have degradation of them. When the signal is not there, usually they do get rid of it. Or you can also think of it in the other lines. A signal is bound to the receptor. How long should it be bound to the receptor? What if it is constantly, the receptor is kept on? That will keep influencing the doer, and we'll have gene expression continuously on. Similarly, the doer, anyway, as we discussed here, it is regulated often. So, but there should be a way to turn off the signal. We always read about turning on the signaling pathway. Okay, what mechanisms are there to turn off? And eventually, when the genes are turned on, there should also be a way of how to turn off the genes, isn't it? Why I'm discussing all these things is, as we said, in stem cell, the signaling pathways that we are going to learn are involved in normal cell signaling pathway, stem cell signaling pathway, and also involved in cancer stem cell pathway. And if we want to treat cancer stem cells, probably we can try these routes, how to inhibit these signaling pathways by blocking any of these steps. So the question is, which is the best place to interfere if you want to? We will come to that question in a later slide. First, we will discuss about Notch signaling pathway. This is an interesting one, and there are few details. And this is a question that usually comes up in the exam, and it will be good if you can practice drawing the diagrams. Okay, by now you should know that I don't like that you write essay questions. I would rather like demonstrate your understanding or memory or recalling through diagrams and other things. Okay, schemes probably, or flowcharts. So practice this. Don't think that without practice we can directly do it in the exam hall. It's an important question. All the three signaling pathways, they are important. Try to do it. Okay, so this is the notch receptor. Okay, remember this, we always had signal, then we said receptor, and then we discussed about doer, and then the genes that are turned on. This is usual cycle. There could be some other steps in between here and here. Okay, there might be some other factors also. I am not writing. Wherever we require, we will probably discuss them. Let's see how this notch signaling path, notch receptor is produced. Notch is produced as a single polypeptide. That means it is continuous with N terminus to C terminus. Okay, it is one single peptide, and it is produced into the endoplasmic reticulum, rough endoplasmic reticulum, ER, and then it will form vesicles and move into the Golgi complex, where it will receive the there will fucosylation and glycosylation will happen. And there is something that will happen also in trans Golgi, is that there is a protease called as furin-like protease, which will break this polypeptide into 2 bits, but it is still bound to it. Okay, now it is something like this. I'm drawing it linearly, but you should understand that they are now interacting too. Here it is, in the first one, it is a continuous peptide. That means several amino acids are bound to it. Whereas in this case, it is a non-covalent bond between the top and the bottom chain. Okay, it is cut, that the protease cuts the notch receptor into 2 peptides like this. Okay, and then it will form a vesicle and will be exposed on top of the cell surface. And I want you to recollect a few things. Addition of oligosaccharides and everything happens in the Golgi complex. Those parts that are exposed into the cytoplasm usually do not have these carbohydrates or oligosaccharides on their surface. It is either into the endoplasmic reticulum or Golgi complex or towards the extracellular side. Okay, you can remember it that way. So in B, at the stage of B, there are 2 important things. One is the oligosaccharides are added, and then the second is the protease, furin-like protease, cleaving the single polypeptide chain into 2, but they are still bound together. So there are 3 parts to the notch signal, as we can see here. This is the extracellular side, and this is the intracellular side. And you have a small region here, that is the transmembrane region. Right? I'll try to draw it here. Assume this is cell membrane. We have a region that is extracellular side. We have the intracellular side as well. Okay, and this is the transmembrane region. Transmembrane region should be having a lot of hydrophobic amino acids, just because the interior of the cell membrane is hydrophobic tails we have. This is the extracellular side, and this is the intracellular side, right? Now, there is a cell here in the top one, if you can see here, and that is producing a ligand, which you call it as notch ligand. It is ligand for the notch receptor. If it is there, it will be bound to the notch signaling on the extracellular side. And if that binding happens, there will be conformational changes, because of which there is another protease, which is called as S2 cleavage. Okay, I forgot to mention, this is called the first cleavage by furin-like protease is called as S1 cleavage. And upon notch binding to its ligand, there is something called as second S2 cleavage by ADAM protease. ADAM protease will cleave on the extracellular side here, somewhere here. And now what we are left, the extracellular side is usually taken up by the cell, and some other processing is there, which we are not concerned about at this time. So upon losing the extracellular domain of the notch signaling. What is left is the transmembrane region, and then we have left with the intracellular side, right? And then there is monoubiquitination. That is another type of post-translational modification, which usually is polyubiquitination, would usually end up with degradation or marking it for degradation. Proteases will bind and degrade the whole protein. Here, this is kind of monoubiquitination. So it has a different meaning, which is to form, it will form an endocytosis, meaning that this whole cell membrane that is present here will invaginate and form an endosome like this. So the Notch receptor that was actually present on the cell surface is now in a vesicle, endosome, right? And there something else happens that is called as S3 cleavage. In S3 cleavage, there is gamma secretase. Gamma secretase will cleave this intracellular domain and leave it. This is the intracellular notch domain. It is also called as notch intracellular domain, NICD. Okay, notch intracellular domain. This notch intracellular domain will now be translocated to the nucleus. This is the nucleus, and there it will act as an activator. Okay, so that is how the notch signaling works. Let's have a quick review of what we have seen, and I also want you to discuss in the way of what is the signal, what is the receptor, what is the doer, and what genes are expressed. So there is some degree of memory that we have to mug up a little bit of it, and that is all right, I think. Okay, so let's see what is the signal here. Yes, anybody? Signal would be the notch ligand. What is the receptor? Receptor is the notch receptor that is present on the cell surface. What is the doer? Doer is also part of NICD, part of notch receptor, but it has been processed, and then they allow the active expression of genes involved in proliferation and other things. Okay, so what you have to remember in this case is S1 cleavage, S2 cleavage, and S3 cleavage. S1 cleavage happens during the processing of the receptor. S2 cleavage happens after binding to ligand. S3 cleavage happens after endocytosis, and then we get NICD. Right? You might also want to remember the names. S1 cleavage by furin-like protease, S2 cleavage by ADAM protease, and S3 cleavage by gamma secretase. Okay? Yes. Any questions here? These are some of the things important, especially trying to visualize how can cells sense the surroundings. That's a very nice way. Cell has the potential to, there could be many different types of signals around, but cell can produce based on what it can, based on what it already is, differentiation lineage, and then respond to the appropriate signals and become one, modify itself or its properties. Okay, notch ligand. Yeah, this is another picture of the same, so I will skip it. So the other one is called as the Wnt signaling. I forgot to mention is one more thing, the name of it. Notch signaling is called notch signaling, and the name is derived from the receptor. Right? Receptor is notch. The name, we call it notch signaling. It's based on receptor, but also it also acts as the doer as well. Notch is special in that sense, but there is processing that is required for notch to become from receptor to become as a doer. It needs several processing steps. There is another one that is Wnt signaling, and it is dependent on, the name comes from the signal. The signal molecule or the ligand is called Wnt. There it is. Okay. In Wnt signaling, let me see if I can zoom in. Excuse me. Okay. So this is notch signaling, Wnt signaling. It is based on the ligand Wnt. If Wnt is present, or you can talk of it as Wnt is absent, what would happen? The receptor for Wnt is called as frizzled. Okay. And frizzled will interact or activate disheveled. Okay. These are crazy names, but DVL you can say. And that will allow the stability of beta catenin, and beta catenin will be carried, will go into the nucleus and will activate transcription of several genes involved in adhesion, reduction in adhesion, and increase in migration. That means it will help in metastasis as well in cancers. Okay, we have to revisit this again and again because we'll have to continue with what is happening. Let's break it down into Wnt is the signal, result is the receptor, and beta catenin, the one that is actually going into the nucleus and acting as a transcriptional factor or activator, is beta catenin. So why is beta catenin not there? What would happen if Wnt signaling is not present? Wnt signal is not present or ligand is not present. When Wnt is not present, it is inhibited, frizzled is inhibited by SFRF, because of which DVL is not active. If DVL is not active, there is something that will happen is there is a whole complex of GSK, axin, and so on, which will which will signal the polyubiquitination of beta catenin. As I said in the previous one, polyubiquitination, that is adding multiple ubiquitin moieties onto a protein, then the protein is targeted for degradation by proteosomes. So beta TRCP will polyubiquitinate beta catenin and result in degradation of the beta catenin. Here, as I said, DOR is controlled by two mechanisms. One of it is, the first one is by stability, and the second one is by the protein or modification, right? Post-translational modification. In this case, if you look at this, normally beta catenin is rapidly degraded. So beta catenin is unstable in those circumstances. Whereas when Wnt is present and then DVL is active, and DVL will inhibit several other proteins, including those of GSK, CK, from acting on beta catenin, because of which beta catenin is not polyubiquitinated, because of which you will have higher concentration or higher stability of beta catenin, which will then be translocated into the nucleus and will allow the transcription of several genes involved in migration. Okay, so let's just recollect what is happening. There are, don't worry, be alarmed about several gene names and all those things. I don't want you to mug up all this stuff. That is not the job we have, but it is about understanding the whole concept of it, the grand, what is the whole thing. Otherwise, just I myself won't try to even remember all these names. Okay, and there are, if there are more number of these genes, then you will have more number of other signaling pathways. In the sense, there is highly complex signaling pathways are usually there is a lot of crosstalk between signaling pathways, right? So what I want you to remember at this stage, simplified, is Wnt signal, that is the signal in this case, and the name of the signaling pathway is based on the signal, that is Wnt, and the receptor is frizzled. The doer is beta catenin, and how is it regulated? Based on its stability. And in normal conditions, it is polyubiquitinated by beta TRCP and degraded. But in the presence of Wnt, beta catenin is prevented from acting upon or being polyubiquitinated, so it is not degraded. We have higher concentration of beta catenin, and it will be translocated into the nucleus, and then there will be action on the target genes. Okay. Yes, I will stop here. If you have any questions, we can talk about it. There is yes, tomorrow we will talk about hedgehog signaling and some of the things, how to inhibit them, because as a part of this lesson, we are learning about cancer stem cells to understand the behavior of cancer stem cells, but also to inhibit the cancer stem cells. What I would encourage you to is go through YouTube. There are fantastic new, more and recent updates on Wnt signaling and the signaling pathways. You would want to watch them. I'll try to share these PPTs, all the PPTs, and you might have a look at them as well. Usually, I have more slides than what is actually shown. I put those slides in the end. Those are kind of additional reading, or if you might see it in a different way, you might probably understand differently or better. So I do have more slides in that. You can read them as well. Okay, any questions? We'll stop here. Any questions, anybody?
No questions, sir.
Okay, yeah.